Hello, this presentation will be done on RT-PCR by myself, Gio Garcia. RT-PCR stands for Reverse Transcription Polymerase Chain Reaction. It is a technique that is used to detect and quantitate mRNA. The technique involves two major steps. Step number one is the synthesis of complementary DNA from RNA by reverse transcription, and two, amplification of a specific cDNA by polymerase chain reaction. Reverse transcriptases, or RTs, use RNA as a template to synthesize the first strand of cDNA, which is then used as a template for the polymerase chain reaction. So what are the uses of RT-PCR? RT-PCR is widely used to diagnose genetic diseases, as well as determining how much of a specific RNA molecule is present in a cell or tissue. RT-PCR can also be used in the insertion of eukaryotic genes into prokaryotes. This is done by taking the eukaryotic cell that contains the gene of interest and extracting the fully modified mRNA molecule. The mRNA is then exposed to reverse transcriptase, which produces the DNA molecule that is complementary to that mRNA molecule. The DNA-RNA complex is then separated under basic conditions. The RNA molecule is hydrolyzed, while the DNA molecule remains intact. A primer is then created to form the complementary DNA strand to the newly formed cDNA to form the double helix. The newly formed cDNA molecule can now be inserted into a vector and placed in a bacterial cell, which can synthesize the protein that we desire. Scientists are also using RT-PCR to diagnose and treat cancer. RT-PCR is used in research to measure gene expression and is also used in public health monitoring. This image comes from an experiment that was done for the detection of naturally occurring enteroviruses in water. This was done because water is generally recognized as a vehicle for the transmission of diseases. So pretty much they wanted to know what type of procedure will be best for the detection of viruses in water. As you can see in the image, at the top, the letters A, B, and C, they correspond to the three different procedures that were used in this experiment. A corresponds to direct hybridization, B, cell structure, and C, RT-PCR. Under each panel, you can see the letters TCID50, which stands for 50% tissue culture infectious probe, which is the amount of a pathogenic agent that will produce a pathological change in 50% of cell cultures inoculated. The numbers to the left, 10 to the power of, correspond to the concentration of the viruses in the water. And under each panel, you can see the letters PV3, CB5, and E7, which correspond to the three different viruses that were used in this experiment. PV3 is poliovirus type 3, E7, ecovirus type 7, and CB5, Coxsackie virus B type 5. So for panel A, which is direct hybridization, PV3 caused positive signals with 10 to the 4 to 10 to the 7. CB5 caused positive signals with 10 to the 5 to 10 to the 7. And E7 caused positive signals with 10 to the 6 to 10 to the 7 TCID. As you can see, panels B and C were far more sensitive than panel A. And panel C, which is RT-PCR, was a little bit more sensitive than cell culture, which is panel B. So RT-PCR was the most sensitive procedure, which means that it was the best procedure for the detection of naturally occurring enteroviruses in water. The only drawback that I can see from this experiment is that there was no quantitative results for the RNA present in water. Quick question. If gene insertion is possible through RT-PCR, could this procedure be used to cure any genetic disorder? For example, in the case of cystic fibrosis patients, we cannot produce a normally functioning CFTR gene? Could this procedure be used to synthesize a normal functioning CFTR gene and introduce it to patients with cystic fibrosis to potentially cure their disorder? These are my references, and I hope you guys enjoyed the presentation.